Hello, IPM students. This is the lecture that I gave on Thursday, March 18th. I had a problem with Camtasia. It crashed and deleted the talk I gave then, so I am currently re-recording it. So just as a reminder, to those of you who are watching this before the week of April 4th, there will not be any lab on Tuesday the 4th. That is because I will be in Portland, so I won't be around to actually go to Bayer Crop Science. I spoke with Amber, the pathologist who will be running our lab, and she said that's fine. She didn't have a whole lot to do on the 4th anyway, so she will just move it down to the next time we meet, which will be the week after spring break. However, I will be back on the 5th, so I can take the students who meet on Thursday lab to Bayer Crop Science. We will be having our lab there, and we will meet there. So Bayer Crop Science is at 266 South Monroe Avenue in Fresno. It's basically right off of 180. So you can get on 180 heading west towards Kerman, and just keep following it until you see the intersection for North Monroe. You'll make a left turn, so you're heading south on Monroe, and Bayer Crop Science will be on your left maybe a quarter mile down the road. It's got a big sign, it's very obvious. You can't really miss it. Bayer Crop Science actually borders 180, so it's pretty easy to find. Just as a reminder, if you are going to Bayer Crop Science, you should dress appropriately. This is for your own safety and for liability of Bayer, so they require you to wear long sleeves, you have to wear long pants, and you also need to have closed-toed shoes. So basically you're trying to cover up all of the skin that could potentially be exposed to the sun, bits of dirt, pesticides, odds and ends like that. You are recommended, but not required, to wear sunglasses, hats, and sunscreen as well, just for your own safety. So with that, one last reminder, when we leave on Thursday, I will be driving a van, so if you want a ride, just show up to the lab room, room 119, and we will leave at 2 o'clock. We will all plan to meet at Bayer Crop Science at around 2:30. All right. So with that, I'm going to hop right into the lecture. We're going to skip over all the quiz stuff because at this point you were either at lecture and heard about it, or you're just going to miss out. So, resistance. Today we are talking about not just resistance, we'll talk a little bit about what resistance is, how organisms become resistant to pesticides, but we'll also talk about how to manage it. And I'm only going to cover the material that I got through on Thursday, so we will cut a little bit short of the end of the actual presentation. So, just as a review, what is resistance? And specifically, what is resistance to pesticides? Well, in a general sense, pesticide resistance is the decrease in susceptibility of a pest population to a pesticide that previously controlled it. So essentially, you have some pesticide or some control tactic that you've been applying for a while now, and over time you notice that it's less good at controlling the pest. The pest seems to be able to overcome its effects, so that you get less control and you end up with more pest population and possibly more damage. So this is all a function of natural selection, because whenever we apply a control tactic, we favor individuals that are less susceptible to it. So in essence, what this means is that we have a population of pests, right? So a group of pests that all live roughly in the same area and can interbreed with one another. And within that population, we're mostly going to have a bunch of susceptible pests, or in other words, pests that are capable of being killed by the pesticide. And the reason that most of them are susceptible is just that no pesticide company is going to market a chemical that doesn't kill most of the pests, right? But within that population, there typically is going to be at least a handful of individuals that are resistant to the pesticide, meaning that the pesticide doesn't kill them as well. They may be 100% resistant, so that you spray a huge quantity on them and they survive, or it's possible that they're just a little more resistant. Maybe they are 10, 15, 20% less susceptible than the susceptible individuals. But either way, you spray your pesticide, and after the exposure, you end up with a reduced population. There's going to be a handful of susceptibles, those that maybe migrated in, maybe they just were in such a place that they did not encounter the pesticide, or they got a sublethal dose. But either way, you always have some susceptibles survive. And then you'll also have the resistant individuals. The 3 that are not susceptible to the pesticide will always show up. So consequently, this is going to be the population that mates and forms the next generation. And since now we've shifted from having a minority of resistance to a majority, when we look at the next season's population, there's going to be more susceptible individuals than there were before. So when we do our spray, we're going to have slightly less effectiveness. We're still going to have our handful of susceptibles, but because there were more resistant individuals to begin with, they're going to compose a larger chunk of the new population. So you repeat this over and over again, and eventually over time, you'll get to a point where actually the majority of the population is resistant. And so it doesn't matter how much pesticide you apply, it's not going to be very effective, because you're just killing a minority of the population that is susceptible. Hence, resistance. Now, we're talking about resistance in terms of pesticides, because that's really where most of the concern with resistance is. But I just want to say that resistance can occur in any system, not just pesticides. There are cases of resistance to naturally resistant host plants. So cultivars that are supposedly resistant to pests can eventually lose their ability to control. There's resistance to biological control. Certain aphids, for example, carry around bacteria that can be used to kill parasitoid eggs before they hatch. And there's even resistance to cultural techniques, like crop rotation. There are certain pests that can learn to lay their eggs in crops that will be rotated to their hosts the next year. So there's lots of ways to get around this. Pests are really good reproducers. They produce lots of generations. They end up generating a lot of genetic variability. So they're very capable of developing these resistant genotypes or phenotypes. All right, so one last thing I want to point out before we move on is this idea that resistance is pre-adaptive. And by that, I mean that in order to be resistant, you have to inherit a gene from your parents, or you have to gain it through a random mutation. And the main point I want to make with that is that there's this general misconception that you get when you talk to some people, that they are under the impression that, well, it's this general misconception that if you expose an insect to an insecticide or a fungus to a fungicide or whatever, that essentially the insect will undergo a mutation event that causes them to develop resistance. That it's sort of a cause and effect, right? That the pesticide causes the pest to mutate into a resistant form. And this is just not true. Either the insect is carrying the trait or it's not. Just by spraying them, you're not going to cause the individual to change, you cause the whole population to change in the frequency of resistant individuals, right? So essentially what I'm saying is, it's a numbers game. That either there's a random mutation that happens at some point and it's unaffected by the pesticide, or that resistant mutation already existed and the parents had it and passed it on to their offspring. Now, resistance is increasingly becoming a big problem in agriculture all around the world. And this is especially true when you're in areas like California or the developing world, where they are highly dependent on chemical pesticides for the control of their pests. This is an especially pronounced problem in insects. There are over 500 insect species that are known to be resistant to at least one insecticide. There are many species within this that have resistance to multiple insecticides. Weeds are about half that at 250. And while fungicide resistance is somewhat rare, it is increasing, and there are certain fungi that are really good at developing resistance, like Fusarium. There's very few fungicides that control Fusarium, and the ones that we have oftentimes break down very quickly. Overall, you can see here that this is a problem that's growing very quickly. It appears to have started out as sort of an exponential curve, but now it's sort of tapering off to more of a linear trend. But as you can still see, we're picking up somewhere around 500 new cases of resistance about every 20 years. So maybe about 40 every year cases of resistance are emerging. And this is especially pronounced, as I mentioned before, in California, where there have been 77 species reported. This is basically a third of all U.S. cases. And a big reason for this is just that we are so dependent on chemical pesticides. And a big part of that is that we grow a lot of specialty crops. We have things like oranges, almonds, peaches, nectarines, pears, apples, whatever. Basically, lettuce as well. Essentially, crops that aren't processed in such a way that we can hide blemishes, but rather are bought at the supermarket at face value. And so people can be a little picky. And they can say, I want to have lettuce that doesn't have any holes in it. I want a peach that doesn't have any bruises, blemishes, or scars, right? And so there's a lot of pressure on growers to produce blemish and damage-free crops. And that typically requires more pesticides. So, how do pests actually evolve resistance? We oftentimes talk about pests just having resistance, but how do they actually do it? What is the mechanism? So we're going to go through those very briefly. The first one is biochemical resistance. And this is by far the most common resistance mechanism in insects. It's a little less common in weeds and in fungi, but again, this is very common in insects. And essentially what it is that insects carry around these enzymes that are capable of detoxifying pesticides. And so what they will do is they will essentially increase the amount of these enzymes they produce, or they will make them more efficient, so that when the pesticide enters the body, these enzymes will detoxify it before it ever reaches the site of action. So essentially, a pesticide has a specific location inside the pest that it needs to bind to. Say you're an organophosphate, you bind to the acetylcholinesterase inside the nerves of the insect. And so what the insects would do is produce a whole bunch of enzymes that will encounter those organophosphates and break them down into a non-toxic form before they ever get to the nerves, therefore protecting the insect. And typically the way this is done is that, like I mentioned, they just upregulate the amount of enzymes they produce. So for example, cotton bollworm, these guys developed pyrethroid resistance decades ago, and a study demonstrated that the way they did this was that they just upregulated their cytochrome P450 detoxification enzymes about 433 times, so that resistant individuals are basically cruising around with about 433 times more detoxifying enzymes than their susceptible relatives. And generally, these are expressed constitutively, meaning they always are expressing this massive amount of detoxification enzymes. And that's because a lot of pesticides are very fast-acting, such that if you get sprayed, death often takes place within 1 or 2 hours. So that's not really enough time to start turning genes on and producing detoxifiers. You need them on all the time. Another approach that you can take is, like I mentioned before, pesticides have a specific site that they target in order to cause the toxic reaction. Another way to get around toxicity is to modify that target site so that it no longer actually fits the pesticide and you don't have that toxic reaction. So you could kind of think of it like a lock and a key, right? A pesticide is like a key. It's going into the pest's body and it's searching for the lock it fits into. And when it gets into that lock and turns, it causes a toxic reaction which kills the pest. So what the pest can do is they can modify the lock so that the key no longer fits in it. And that way it can't turn the lock and it can't cause the toxic reaction. Alternatively, what the insect can do is it can produce a lot fewer locks so that it's harder for the pesticide to bind to enough of the locks to cause the toxic reaction, therefore protecting itself that way. This is a rather common tactic that's used in insects, fungi, and in weeds. A really nice example of this is in Pyrenophora teres. This is a common sort of blight fungus in, I believe, barley is primarily where it's found. And so it has resistance to demethylase inhibitors. These DMIs are a pesticide that bind to CYP51 enzymes. And these are enzymes that are essential in basically converting carbon into sterols. Sterols are sort of the base molecule used to make steroid hormones. And so without those, the fungus can't survive. It can't send signals around its body. Now what P. teres did was they modified their CYP51 so that the active site was really narrow, and it could basically only fit these sterols through a very specific conformation. And the DMIs no longer could fit inside that active site. And so essentially the keys no longer fit the lock, and the fungus could grow uninhibited. Now, something you see in plants, as well as in insects to a certain extent, is the use of sequestration. Now, in this approach, the plant or insect or whatever pest you're looking at, it doesn't really bother with modifying any part of its body. Its idea is that there are certain tissues that are vulnerable to the toxin, and there are certain tissues that are not. And so what the pest will do is they will sequester the pesticides to a part of the body that's not vulnerable, so that those toxins never ever get to the vulnerable tissues. So a nice example of this is in glyphosate resistance, Roundup. So Roundup is widely applied across more or less all weed systems, as long as you have Roundup ready crops. And resistance has been increasing over the years. And one of the big ways we see this working out is with sequestration of glyphosate into vacuoles. So the idea is that glyphosate is toxic by kicking photosynthesis into overdrive and burning the plant out. So in order to have the toxic effect, it needs to enter the chloroplast and start messing with the photosynthetic machinery. So typically glyphosate gets sprayed, the glyphosate mobilizes into the, well actually I'll explain this with the graph. So what we've got is a graph here showing where glyphosate goes inside the plant after the plant has been sprayed. So in black, we have the vacuoles, or storage containers in the cells. In red, we have cytoplasm, or the spaces in the cells. We have the chloroplasts in green, and then in yellow, we have mobilized, meaning it's in the intercellular space or in the vascular system moving around the plant. So in a susceptible plant, what you can see is that right after the spray, in time, within about 2 to 4 hours, most of the glyphosate has entered the cytoplasm, so it's inside the cells. But then very quickly after that, it starts entering into the chloroplasts, and then there's this mass mobilization. A lot of it goes into the intercellular space or the vascular system, and it all gets moved around the plant and eventually put into chloroplasts all over the place, until almost all of it is in the chloroplasts, starting to burn out the plant. Now, in the case of resistance, which we have on this bottom graph, what the plants do is that they overexpress cell-based transporter proteins. So these are proteins inside the cells that basically grab molecules and transport them to new locations. So they create a whole bunch of these little transporting proteins inside their vacuoles, so that when you have that initial spray, a lot of the pesticide, the glyphosate, goes into the cytoplasm, but instead of having this huge mobilization that moves it all over the plant, most of it gets pulled out of the cytoplasm and put directly into the vacuoles. So if you remember your cell biology, the vacuoles are just great big holes in the middle of the cell that are used for storing materials. There's no chloroplasts in them whatsoever, so the glyphosate is safely stored in a location that is far away from the chloroplasts. Additionally, you see very little to none of it is mobilized, so it's not really moving around the plant, going out to the growing tissues. Instead, it's just kind of sticking around in the cell and the places that got sprayed directly. So some of those leaves will die because a certain amount will get into the chloroplasts, but the glyphosate won't spread in the plant. So it's not going to reach all the meristems, it's not going to hit all of the cells that are dividing, and so the plant can survive the spray and will just keep growing. So again, sequestration is essentially taking pesticides, putting them in a part of the body like the vacuole, where they are non-toxic, and just keeping them away from those vulnerable growing tissues. And finally, as far as changing physiology, there is also the ability to reduce the penetration of the pesticide. So, before the pesticide can have a toxic effect, the first thing it needs to do is it needs to actually penetrate the pest. So it needs to get through their exoskeleton, through their cell walls, through whatever they have, in order to get into the body and have their toxic reaction. So if the pest can modify its external anatomy so that there is less of an ability of the pesticide to get in, then they can be resistant that way, because they are just getting a lower dose. This has been found in bedbugs. So bedbugs for a long time were really well controlled with a handful of pesticides that could be used inside, such as pyrethroids. Pyrethroids are generally pretty safe to people. They're generally safe to pets, except cats. And so, overall, there's very little danger from spraying pyrethroids in a house, and so they were used largely for bedbug control. However, in the early 2000s, bedbugs developed resistance, and so we had sort of this huge bedbug outbreak that is still ongoing. And that's why all of a sudden it seems like bedbugs are a bigger problem than they were 10 years ago. As it works out, there's a couple mechanisms bedbugs use, but one of them is that resistant populations have evolved this extra thick, waxy layer on their cuticle, right? So insects have a chitinous exoskeleton, but on top of that exoskeleton is a layer of wax that is designed to basically keep water inside, but it also serves the purpose of stopping pesticides from getting into the insect. So there was an experiment where some researchers basically collected a bunch of bedbugs and exposed them to a glass plate that was covered in pyrethroids. And they measured how long it took the bedbugs to die after walking across the plate. And they basically split them up into 3 groups: those that were intolerant or died in less than 2 hours, those that were tolerant, survived longer than 4 hours but less than a day, and those that were resistant, or didn't die within 1 day of exposure. So they took these bedbugs, and they looked at how thick their wax cuticle was, and they saw this really nice linear relationship, that as the cuticle got thicker, they developed more resistance. And they hypothesized that this was because it was harder for the pyrethroid to get through this nice thick cuticle. And then in addition to this is sort of a rare case, but it is possible that pests will develop resistance through something called excretion. So in excretion, the pests essentially just do exactly what it sounds like. They excrete the pesticide from their body in an unmetabolized form. So they don't detoxify it, they don't sequester it, they just let it pass through their bodies and, basically poop or pee it out without any toxic effect. This works pretty well for pesticides like Bt that are non-toxic in their standard form and have to be digested by the pest into a toxic formation. And so in that case, a pest could say ingest Bt and then just never metabolize it, and that way it never has its toxic effect. However, through my studies, my impression is that this is a really rare defensive mechanism, and generally it only occurs in pests when they're fighting sort of natural defensive compounds in plants. For example, tobacco hornworm can feed on tobacco and ingest massive amounts of nicotine, which is toxic to most insects, and excrete it unmetabolized. But, then again, there's very little evidence of this mechanism being used against pesticides. And then lastly, it's possible for insects, and I believe just insects. I don't think you'll get this with fungi, pathogens, or plants because they don't really have behavior. But it's possible for insects to change their behavior so that they limit their exposure to pesticides. Essentially, there's a pesticide in some location, but the pest just naturally doesn't go there and get exposed. Just sort of like in this picture, if you spray a plant from the top down, you are going to basically kill any pests that choose to walk across this upper surface, but if the pest preferentially spends time on the undersides of leaves, it may be safe. This is a surprisingly common form of resistance. For example, tobacco budworms, whenever the larvae are feeding, if they encounter pyrethroids, what they'll do is they'll basically just stop feeding for a while. They won't move, they'll stop feeding, and they just will decrease their exposure for a couple of days until the half-life is basically broken down, all those pyrethroids. Similarly, diamondback moths, these guys, they're sort of masters of resistance. They're resistant to like 8 different modes of action of insecticides. They actually show preference in where they lay their eggs. They seem to be able to detect if a leaf has been sprayed or not with various insecticides, and they will avoid laying eggs on insecticide-sprayed leaves and will instead target untreated ones, so that when their larvae hatch, they will have a nice clean source to feed on. So those are all the mechanisms that are associated with resistance. And before we move on to actual sort of resistance management, there's a couple other topics I'd like to cross and to talk about. And the first of which is cross or multiple resistance. And cross or multiple resistance is the idea that pests can have the ability to be resistant to more than one pesticide at a time. So they may not just be resistant to, say, pyrethroid, but they may also be resistant to organochlorides, right? And there's 2 major ways that this can happen. The insect could have what we call cross resistance. By insect, I mean pests generally. This doesn't just apply to insects. But the pest could have what we call cross resistance. And this is where the pest has a single gene mutation that provides resistance to more than one pesticide. So essentially, one mutation, multiple resistance patterns. Generally, what we see is that this resistance is going to be against pesticides in the same class. So if you have cross resistance to, say, one pyrethroid chemistry, then it's very possible that resistance mechanism will give you resistance to most of the pyrethroids, because pyrethroids all share the same mode of action. So detoxifying one pyrethroid probably will also detoxify all the others. Alternatively, you may have different modes of action, but they may target the same site of action. So if your defensive mechanism is one of those modifications of the target site, then you might get resistance across modes of action. For example, there's a really well-known resistance gene called knockout or knockdown, which is found in a wide variety of insects. And essentially what it is it's a modification of the sodium-gated channels inside the insect's neurons. And since sodium-gated net channels are targeted not just by pyrethroids, but also organochlorides like DDT, this knockdown mutation provides resistance to both organochlorides and pyrethroids, even though it is just a single mutation. On the flip side, we have what we call multiple resistance. And this is when a pest is resistant to multiple modes of action, but it does so through having multiple unique resistance genes. So essentially, you may be resistant to organochlorides, organophosphates, and carbamates, but it's not because you have a single gene mutation that gives you detoxification of all of them, but rather you might have 3 different genes, one for each individual class of insecticide. And this is probably the most common way that pests get resistant to multiple pesticides, and it's typically because it's the way we oftentimes treat pesticides. We spray them over and over and over again until resistance develops, and then we switch over to the next pesticide. And so basically what you do is you say, let's say you're using organochlorides like DDT. You spray DDT over and over and over again until it doesn't work anymore because your population is resistant to DDT. Then you move on to an organophosphate, and you spray those over and over and over again until you have resistance to that. But because the original population you were spraying was already resistant to organochlorides, you're basically selecting for individuals that are resistant to organophosphates while already being resistant to organochlorides. So this is, like I said, sort of the common way that you find resistance. There are certain insects that are really good at accumulating resistance. For example, diamondback moth has 8 different modes of action that it is resistant to. You see similar levels of resistance to things like in spider mites. They're highly resistant. So are the some aphids, like the cotton peach aphid or the melon aphid. They have a really high resistance rates. Now, no. Now, you might be thinking, but Dr. Wenger, if there are these genotypes, right, that allow pests to be resistant to pesticides, why aren't all pests resistant? Because you would think these resistance traits are going to be hugely advantageous. They're going to benefit the insect or the fungi or the weed or whatever. So why don't they spread really quickly? Why don't pests just naturally have resistance to pesticides? Well, there's a perfectly logical evolutionary reason for this, and that is that most resistance genes have something that we call a fitness cost. And essentially what I mean by that is that resistance alleles, or in other words, gene variants that cause resistance, typically don't work as well or as effectively as the natural versions of those genes that occur before resistance evolves. So if you think of it, our genes and our proteins, as well as the proteins of pests across the board, are the product of millions of years of evolution. Essentially, generation after generation after generation, selecting for the very most efficient genes, the very most efficient proteins that do the job with the least amount of energy the most effectively. And those particular genes, those particular proteins, are susceptible to our pesticides because we are trying to target a mechanism that is essential for the pest. Now, whenever we have a mutation that provides resistance, we are basically taking one of these highly efficient proteins and we are modifying them into a form that is different from that most efficient one. We are essentially making them less efficient. So by doing so, the pest is going to have to expend some extra energy in order to have resistance, because they're not going to have a version of the gene that is ideal. And oftentimes, the cost of having this resistant allele is that they have to spend more energy just to survive than a susceptible pest does with its original genotype. So think of it in terms of that cotton bollworm we talked about earlier. The one that produced 433 times more detoxifying enzymes than the susceptible bollworm did. Now, that bollworm has a real advantage when there's a pesticide in the field, right? Because it's got all those detoxifying enzymes going all the time. But the cost of that benefit is that they have to spend 433 times more energy on detoxification than their susceptible cohort does, their susceptible relatives. Those susceptible relatives are taking that energy, that 433 times resistance energy, and they're applying it to other things. They're using it to grow faster. They're using it to produce more eggs. They're using it to find mates, to eat more, right? They have more energy. So, if you were to remove the pesticides from the equation, then those susceptible individuals would actually do better than the resistant ones because they have more energy available to them. They can go out there and reproduce faster. And over time, without that pesticide resistance, we expect those resistant genotypes to be less adaptive and to decrease in frequency because they're going to mate less. They're going to survive less. They're going to produce less eggs when they do mate. And so overall, the general idea is that as long as you're applying a pesticide that favors a resistance gene, then you're basically reducing its fitness cost. You're actually giving it a fitness advantage. But as soon as you remove that resistance as soon as you remove that pesticide, that cost becomes obvious and the frequency is going to decrease. And that's why natural populations that are not exposed to pesticides tend to have really low resistance frequencies. Consequently, this is part of the the entire theory of mode of action rotation. is that when you rotate a mode of action, not only are you reducing the selection pressure that favors the pest, but also you are reducing the amount of time that the pesticide is in the field. So you are reducing the amount of time that the pest resistance trait is favored, and you are increasing that cost of having that trait. So, general gist, resistance costs energy, it costs growth, it costs reproduction. So it only increases if we are selecting for it with sprays. So, we know that resistance is caused by exposure, by selection, causing sort of a natural populations to evolve towards resistance. But there are also certain biological factors of the pest that can cause the resistance trait to evolve more quickly. In such cases, essentially we expect certain types of pests to become more resistant more quickly than others. And some of these traits, well, essentially all of these traits are associated with aspects of the life cycle that make the pest have greater exposure to the pesticide and more selection pressure acting on them. So first off, resistance tends to occur more quickly in species that are short-lived. And the reason we say this is that essentially they have a rapid life cycle. Short-lived species tend to hit their reproductive age sooner, they lay eggs faster, and they produce the next generation sooner. So you'll have multiple generations per season and possibly multiple exposures to the pesticide per season. Additionally, we see that rapid development is also associated with faster resistance. Now, to be clear, these two operate through a similar mechanism. They both produce more generations per season. But something that differentiates a rapid developer from a short-lived species is that rapid developers spend less time in their vulnerable life stages. So they are a little bit less likely to be super exposed at the key moment, and so you're more likely to see resistance problems just due to a lack of exposure. And finally, we see that resistance builds up most quickly in pests that are relatively immobile. This is because sometimes you spray a pesticide, and pests will just flee the area to avoid exposure. And so you have a large number of susceptibles living elsewhere, not contacting the pesticide. But on the flip side, immobile pests can't leave. They have to basically stay there and stick out the pesticide. And so it kills off a very large chunk of the susceptible pests, leaving only the resistant ones behind to mate. So consequently, when we're looking at things like insects, the pests that tend to evolve resistance the fastest are the ones that fit this mode. They reproduce quickly, and they are immobile. So things like mites, aphids, and scales oftentimes are the worst case scenarios as far as resistance is concerned. Now, in addition to biological factors, there are many production factors or operational factors that can influence how quickly resistance develops. So these would essentially be factors that are associated with how we apply pesticides and how that may or may not encourage resistance to evolve. So generally, the trend is that we tend to increase and select for resistance if we apply the same mode of action over and over again repeatedly. Now this one's kind of a no-brainer. If we spray the same mode of action every time we have a pest problem, of course we are always selecting for the same resistance genes. We're going to increase resistance in our populations. Now, another factor that has an influence is also the persistence of the pesticide. So generally, if the pesticide is highly persistent or has a slow release format, then we're going to increase the amount of exposure the pest has. We're going to kill off more of the susceptibles than a short-lived pesticide will, and then we'll end up with a higher frequency of resistance at the end of the season. Pesticides being applied directly before mating is problematic. The reason for this is that after you apply a pesticide, there's only so much time that will pass before essentially the pesticide has detoxified itself and the agroecosystem becomes a place where the pests can feed once again. So if you were to, say, apply the pesticide early in the season before mating occurs, then you have plenty of time for susceptible individuals to migrate into the field and to establish themselves and to dilute out the effect of resistance. However, if you were to spray, 1 or 2 days before the big mating flight, then when that mating flight occurs, there's no time for susceptibles to fly in, and the vast majority of the population is going to be resistant. Well, not the vast majority, but you're going to have a high proportion of resistance. If you treat every generation, then you're going to get resistance pretty quick. That's kind of a no-brainer. It sticks with the idea of sort of the same mode of action and essentially the persistence, just that you are exposing the pest a lot. If you spray the entire field or the entire pest population. So for example, if you have a field with aphids in it, aphids tend to have hot spots where they're very prolific and causing trouble. If you were to, say, spray just the hot spots with sort of a targeted approach, then you're leaving a fair number of susceptibles in the rest of the field that could move in and mate with the resistant individuals left behind. On the flip side, if you sprayed the whole field, then you're not only eliminating your hot spots, but you're also eliminating your susceptible reserve. Along those same lines, if you were to apply the same pesticide over a large geographical area, you have the same problem, but on just a larger scale. So for example, if you're in an area producing a whole bunch of almonds, and everybody's producing almonds all around you, and you're all applying the same pesticides, then you're going to see more of a resistance problem more quickly, because you'll have resistant individuals in all the fields, not just yours. Also, resistance if you use a low treatment threshold. So essentially if you're spraying when there are very few individuals in the field, again, that ties into the idea of all of these up here at the top, the idea that you're exposing the pest to the pesticide a lot. All right. So, knowing what resistance is, and knowing, So knowing what resistance is, and knowing the sorts of things that cause it to occur, how do we actually manage resistance? Well, there's two major schools of thought. The first of which is reactive management. And reactive management is essentially you treat with the control tactic over and over and over again until it's no longer useful, and then you trust that there's going to be some new control tactic coming along that's going to be useful against the pest. Now, this is effectively no management. You are managing your pests, but you're not taking any steps to prevent resistance, and you just kind of hope that there's going to be something down the line. And this is what's happening more or less universally, especially in cases where there are relatively few modes of action available to growers. So Dave Marler talked about this when he came in and spoke to us in lab. He mentioned that one of the big problems we're facing right now with insects is that there's a lot of resistance and there's very few modes of action. So you can apply, so essentially when a grower finds a chemical that works really well, they'll just use it over and over and over again, because there's no guarantee that they're going to find another chemical that works nearly as effectively. And so you have something like Movento, which is highly effective against mealybugs in grape. You apply it every single season. And as such, maybe 7 years ago, you would get 2 or 3 years of control worth, but nowadays, you're applying it 2 or 3 times a season just to get that same level of control. Whereas on the flip side, reactive management is less common when you're controlling for pests that do have lots of options for control, like fungi and weed species. As Dave said again, there are lots of pesticide modes of action that are useful in those areas, and so they will apply those instead, and they will rotate their modes of action instead of just applying the same thing over and over and over again. Now on the flip side, we have preemptive management, which would be like we saw in the weeds and the fungi that Dave was talking about. This is a case where we alter the design or the execution of our pest management in order to extend the lifespan of the control tactic. Now, the disadvantage here is that ultimately it's going to be more work. The growers are going to have to invest more time and energy into applying different chemicals or possibly using different approaches like biocontrol or cultural techniques. Pest control professionals need to do a lot more scouting. They need to investigate a lot of different techniques. But on the upside, in the long term, you're going to face less resistant pests. So those techniques that you spend time learning how to do, chances are they're going to work longer. So in the long run, you do less work as far as learning new techniques. So really, the difference between reactive and preemptive management is sort of a trade-off in short-term convenience versus long-term benefits. That it is inconvenient to spend this much time trying to manage resistance, but on the upside, you have much less of a chance of these short-term sort of disasters happening where, say, you have a resistance problem and you don't have a tool to fill in. All right. So I think what I'll do is I will take it off from there. I believe in lecture we got a little bit through reducing selection pressure. What I'll do is I'll record this in the next lecture, and I'll leave you right here for now. So thank you for listening. I will see you in the next lecture.